Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Using Real-World Evidence to Enhance Drug Development, Arterial Health Monitoring Innovations. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speaker throughout the presentation and we'll try to attend to as many as possible. If you're joining us via GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And if you're viewing this presentation from our LinkedIn Live event, you can submit your questions using the comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank CardiX who developed the content for this presentation. CardiX is a digital health technology company that develops and markets non-invasive patient monitoring technologies for assessing cardiovascular health. Their pioneering SphygmoCor vascular biosensing technology measures arterial health by collecting clinically meaningful digital vascular biomarkers using novel sensors. These biomarkers represent key indicators of cardiovascular health, including but not limited to central blood pressures, vascular stiffness, vascular age, and heart stress. SphygmoCor has been deployed in multiple clinical trials sponsored by pharmaceutical companies and healthcare systems to measure arterial health. And now let's take a moment to meet our featured speaker for today. Dr. Sanjeev Bhavnani is a renowned physician, digital health clinical trialist, and policy strategist with over 20 years of experience transforming healthcare innovation. He excels at integrating digital health technologies and AI/ML into clinical practice, leading teams into impactful trials achieving FDA approvals. A strong advocate for DE&I, Dr. Bhavnani champions inclusive healthcare solutions and influences national policy to ensure equitable access to advanced medical innovations. And now, without any further ado, I'd like to hand the mic over to Sanjeev to kick off the presentation. The floor is yours.
Thank you very much, Corey. Thank you, X-Talks, the organizing committee, and to our audience. Pleasure to be with you today. Let's get underway. So today's talk is titled Enhancing Drug Development with Real-World Evidence and Clinical Trial Innovations, Arterial Health Monitoring through Connect Pulse. I'm a practicing cardiologist in Southern California, and I'm also a consulting chief clinical officer at CardiX. These devices and trials will be discussed. So let's get right into it. Why do we need real-world evidence and trial innovations? Well, some quick facts. 80% of clinical trials are delayed or closed prematurely because of inadequate accrual. I think many in the audience or those who have participated in clinical trials, whether academic or sponsored, experience this firsthand. There are structural, clinical, and socioeconomic factors that affect trial participation. There is large underrepresentation in trials. And when one looks over the course of the last several decades, less than 10% of the US population, at least here in the United States, has been invited to a clinical trial, and of that, less than 5% follow through with participation. There's a high burden, high cost, and while trials are very well structured and very well intended, they are highly inefficient. So real-world evidence and trial innovations, there are 6,000 hospitals and over 30,000 clinics in the United States. The CDC reports that there are 140 million emergency room visitations. These are healthcare encounters for acute conditions. And over 1 billion physician office visits for wellness, chronic disease management, and post-hospital follow-ups. So, and the number of patient visits to federally funded, that is low socioeconomic clinics, increased from 77 million in 2010 to almost 130 million in 2020. So the point here is that nearly 100% are collecting real-world data through electronic health records, billing, outcome adjudications, pharmacy claims data, and patient registries. So now we have multi-dimensional data as a part of real-world evidence. Can we employ new computational approaches to transform clinical trials? So over the past 10 to 15 years, this mobile technology and digitization of healthcare, in person I would have asked this, but for those in the audience to just think about, have you used a digital health device or some sort of biometric or physiologic tracking device in your own health or somebody that you might know? And really, from head to toe, if you can think of a measurement, a biomarker, a physiologic parameter that can be measured, it is likely being measured by a sensor-based technology. There are smart ring devices to monitor sleep, health, wellness, exercise training, smart necklaces for bioimpedance monitoring in heart failure and pulmonary disease. There was the era of smart pills, sensors in pills to monitor medication adherence remotely, clothing, genome sequencing technologies on USB-sized devices now, and the list goes on and on. So as we look at the emergence and now availability of a lot of different technologies and sensor-based platforms, how do we employ these now with regulatory thinking of employing digital health technologies for remote data acquisition in clinical trials, seeing people in their home or natural environments through data acquisition using devices? Using real-world evidence to support regulatory decision-making for medical devices, conducting clinical trials with decentralized elements, siteless, paperless clinical trials to see individuals and trial participants across geographies through decentralized platforms, and now pushing forward using AI ML in the development of drug and biologic products. I had a unique experience at the Food and Drug Administration to work on these documents and others. So in our agenda today, so we set the stage. So what will we discuss? We'll go over some physiologic measurements, and that's the arterial pulse wave. Very important to know that what you're measuring actually has physiologic connotation and understanding. Digital vascular biomarkers, employing analytical and clinical validation methods in order to look at the robustness of these biomarkers that can be developed. How are they analytically and clinically validated? We'll see some examples of remote patient monitoring, real-world data to generate real-world evidence. And then we'll look forward into precision phenotyping. How do we use a lot and that multitude of healthcare data sets and waveform analyses in order to look at precision phenotyping? And then what are those computational approaches for real-world evidence? So as we get into it, and with blood pressure as our motivating example for today, there are a number of different devices, and they fall into several categories. But there are oscillatory blood pressure devices, the one that inflates, both inflate, but look at different parameters with how we're detecting systolic and diastolic pressures, and oscillometric blood pressure devices. And if you look at the curve on the right, that is a blood pressure waveform. And what we're measuring, right, for those who have measured their own blood pressures or have seen it in other individuals, we're measuring the peak and the trough, the star at the top and the star at the bottom. So you're systolic and diastolic. But one can easily see there is a waveform that is inferring a lot more physiologic information that we don't capture through conventional, traditional blood pressure devices. And so when we look at just conventional blood pressure, and there is no shortage of clinical trials, very large randomized clinical trials over the last several decades that have tackled this question of how intensive a therapy to treat hypertensive individuals to bring their blood pressures down, and what does that mean towards cardiovascular outcomes? So in the SPRINT clinical trial, as one of the many examples, that using an approach of intensive treatment adjustments to bring blood pressure to goal, the intensive treatment arm achieves a systolic blood pressure reduction to 120, where at baseline it is 140, compared to standard treatment, you're every 2 or 3 times a year of seeing a physician for medication adjustments. One can clearly see that there is a method for improving blood pressure quickly and sustaining it. And what that translated to is a 25% reduction in major cardiovascular, that is myocardial infarction, heart failure, stroke, and cardiovascular mortality benefit that happens pretty quickly after 1 year of randomization and follow-up, the curves begin to diverge. So it's very promising, right, in how we look at the safety and effectiveness of intensive therapy on systolic and diastolic blood pressure in individuals with uncontrolled hypertension. But that's not the whole story, right? Well, what happens after? What happens to trial participants after when they now become real world? And so in an extended observational follow-up of the SPRINT trial from 2016 to 2020, a remarkable observation was seen. And that is in this curve, in this straight line here. This is at the end of the clinical trial and observational phase, but entering the real world phase. And that intensive therapy that resulted in blood pressure reduction started increasing back to its baseline. And when you compare it to standard therapy, there was no change at 9 or 10 years after individuals were enrolled in the SPRINT trial. And hypertension is a lifelong condition and requiring decades of therapy. So this change and this resurgence of back to hypertensive state wiped out the cardiovascular benefit that was seen in the trial, that there was no benefit on extended follow-up for myocardial infarction, stroke, or cardiovascular mortality once individuals entered back in the real world. And so this is a little bit of a paradox, right, that I'm presenting to you. But for us to ask this question, why? We know that there are limitations of the usual blood pressure hypothesis. There are variabilities in the real world. There are instabilities in individuals. They don't just have hypertension, they may have other conditions as well. So systolic and diastolic pressure, seeing those stars again on the right, these are point measurements. Trial results are not translatable to real world clinical practice. RCTs to real world evidence. The arterial pulse wave that generates pressure are not measured through traditional blood pressure devices. That blue waveform is inferring a lot more information about the state of the cardiovascular condition in individuals. And blood pressure may change, but is the underlying physiology changing? You can treat and improve blood pressure, but is cardiovascular function improving? So let's take a closer look at this, right? That the physiology of the arterial pulse wave. The animation on the right, right? That this is a mechanical system, but one can see that the pressure wave is moving forward. But as it moves forward, there's a portion right there that starts coming backward. So while one thinks that, the body is a river with blood flow moving in one direction at all times, that's not true. It's moving forward with a pulse velocity, and then it also reflects backward. And this is very important because this is the underpinnings of the cardiovascular system. And that forward pressure that is being generated by the heart is also seeing a reflected backward wave back towards the heart. And so looking again, the pressure wave is moving forward when the heart contracts. And part of that is also reflected backwards. Now the reflected wave and the forward wave, these are the load on the heart. So they are additive. That's an added pressure that the heart has to experience and generate over in order to produce blood flow. And as that reflected wave, if there's more stiffness in the arteries of the body, I want you to think about blowing through a straw that has become smaller in size. You have to generate a lot more force to produce airflow. So the heart has to generate a lot more pressure in order to generate blood flow out into the bodies, the major organs of the body, the lungs, the brain, the kidneys. And it does this on a beat-to-beat basis. So this is the technologies that led to the development of SphygmoCor and AdCor, and now a suite of devices to be able to non-invasively, this had in the past required cardiac catheterization with a pressure catheter directly in the aorta, but through decades of research and validation, that this method is now, through non-invasive non-invasive technologies able to be detected in a blood pressure cuff device. So it's a measurement of the waveform in addition to the systolic and diastolic pressures. And so central blood pressure, the blood pressure that you get when you measure at your arm is a brachial blood pressure or a wrist blood pressure. We'll call that peripheral blood pressure. But as we've seen and as we've talked about, there is a pressure being generated directly at the heart, and that is central blood pressure. And that is what this waveform represents, and the underpinnings of the technology of the SphygmoCor device, as well as now in a wireless and wearable Connect device. And when you look at these waveforms, right, the forward pressure wave, right, is generating this waveform. But how much that pressure wave comes back is known as its augmentation. That reflected wave is augmenting, it's adding to the forward pressure wave. And this is now introducing pulse wave analysis. While measuring systolic and diastolic blood pressure, there is the potential in these technologies that has been validated on these measurements of arterial biomarkers for pulse wave analysis to infer underlying cardiovascular physiologies. So the specific context for use of a biomarker drives the extent of the evidence needed for validation. And I think any topic regarding a digital measurement must include an analytical validation and clinical validation approach. It's not sufficient enough in order to just measure, to create a measurement. That measurement have performance and acceptable characteristics for that biomarker assay. And this comes from the laboratory space and in vitro diagnostics, but applies directly into digital biomarkers. So the reference ranges must be established. The pre-analytical assay performance characteristics. What was the rigor of analytical reproducibility? Is it measured serially and do you get the same results? And then there's clinical validation. Establish that the biomarker acceptability identifies, measures, or predicts the concept of interest. And that concept for us today is going to be cardiovascular physiology, including hypertension. So what was the study design? Was it clinically meaningful? Did it have the right decision points in order to understand how it's used clinically? And then, of course, the benefit risk assessment. And now applying this to the SphygmoCor technologies and to pulse wave analysis, that the normal ranges have been validated over time in large clinical trials and large registries in order to understand in gender and as people age, the differential effects of wave reflections and aortic pulse and on pulse wave analysis. So central blood pressure, much like brachial blood pressure, has a predictable pattern in gender and as time goes on with age. And for central blood pressures, unlike brachial blood pressures, for those who are familiar, it's a near linear association. Central blood pressure, systolic and pulse pressure, go up in individuals as they age. In healthy, normal individuals who do not have cardiovascular disease, goes up as time goes on. And this is true for the arterial stiffness measurements. Augmentation pressure, how much of that wave was reflected back. If there's more stiffness out in the arteries, like blowing through that small straw, right, there is a greater augmentation pressure. And that augmentation pressure also computes an augmentation index that's individualized for that person. But these are taken together to understand that stiffness, arteries become stiff as individuals age. And so in the analytical validation of establishing reference ranges, there is the clinical validation and a multitude of large randomized observational trials and large randomized treatment trials have looked at central blood pressure and its predictive ability compared to brachial blood pressure on major cardiovascular outcomes. And in the Strong Heart study, sponsored by the National Institute of Health, greater than 3,500 subjects, 2,400 who were disease-free at baseline, followed longitudinally and showed that a central pressure greater than 50 millimeters of mercury, with normal being in the 35 to 40 range, so this person has central hypertension, has a 20% increased risk of a cardiovascular event including myocardial infarction, heart attack, heart failure, and stroke in the next 5 years. We do very well for cardiovascular risk prediction. We do generally well for long-term, 10-year, 20-year risk of heart attacks. But what about short-term? Can we understand higher risk individuals through the pressure biomarkers, vascular biomarkers? And what's interesting is that brachial blood pressure did not show a similar threshold. So back to that SPRINT study that we discussed, what are we measuring as far as understanding an individual's risk? And looking at the differential impact of blood pressure lowering, so it's one thing to be hypertensive and have high cardiovascular risk, but can that risk be augmented? Again, clinical validation. And in the CAFE study of looking at conduit artery function evaluation by treating central pressures with either beta blockers and diuretics, or calcium channel blockers and ACE inhibitors or ARBs, looking at no treatment. We understand that no treatment has the same effect in hypertension, but are there groups of treatment that can reduce central blood pressures and then result in improvements in cardiovascular outcomes? And in this study, among hypertensive individuals with central hypertension, they have brachial hypertension, they also have central hypertension. A strategy of using calcium channel blockers and ACE inhibitors was more effective at reducing blood pressure than beta blockers and diuretics alone. And when we look at the not just the measurements of systolic and diastolic blood pressure, but when you look at the waveform and how it changes because of the type of therapy, this curve should become smaller in amplitude, and it should shift towards the right, knowing that pressure is being reduced and stiffness, arteries are relaxing. Calcium channel blockers was associated with a 30% less cardiovascular events due to central blood pressure reduction. So analytical, pressure has normative characteristics and pressures are predictive. And clinical validation that treatment approaches can be employed in order to reduce central blood pressure and understand the evolution and the hopefully regression of arterial stiffness over time. So the pulse wave analysis has been validated in more than 2,500 research papers from head to toe, and have been evaluated and then validated in cognitive function in stroke individuals, where central blood pressure is predictive, but arterial stiffness is also a measure, a treatment target measure, in order to understand individuals' risks across cardiopulmonary disease, vascular disease, renal disease, and even into major cancers such as cancer chemotherapy for colon cancer and breast cancer. So arterial stiffness and central pressure is more than just cardiovascular, right? This is a measurement to understand head to toe body and organ function and dysfunction. So let's use what we've had in the background to look at examples of remote patient monitoring with digital health technologies, real world data to generate real world evidence. So, I'm a practicing physician, and I'll say these are not our patients, right? Many people are not digital natives. Those with cardiovascular condition have a tendency to be older, but these are most certainly not the typical individuals. Others, and I hope that the audience is smiling, that our patients are more like this, right? They can use technologies. They want to use technologies. They understand the value that it brings towards their health and disease modification. But we do need to engage people of where they are for their utility in using a digital technology. So I'll show you some examples of hypertension in some of our own programs and in atrial fibrillation, a cardiovascular condition that is among the highest risk for strokes in the United States. So a smartphone ECG and even smartwatch ECG, for those of you that are wearing smartwatches, this is an example of a 78-year-old individual who has an irregular heart rate known as atrial fibrillation. Right? They may not be feeling well when your heart rate is this fast and this irregular, but we're able to use a technology to monitor this individual at home, right? And then use a decentralized method, a virtual care method, in order to adjust therapy and in this home-based environment. And we can see the conversion of the heart rate that was fast to normal. We can see, this resurgence back to rapid and recurrence of rapid atrial fibrillation. But this is also not just the measurement, this is also conveying risk in a person, right? Are treatments be effective, the ones that are being used? Are they not? Do they need to change? Based on a risk that now is evolving through the acquisition of digital measurements, we can now start asking, what is the right clinical outcome assessment for this individual? And in this person, it was to go for more conclusive therapy for atrial fibrillation in a shorter time period. In what would otherwise require, regular clinical trials and adjudication criteria and time-based interventions, can we start looking at virtual and decentralized methods in order to understand risk in a more real-time approach? Here's an example of a typical blood pressure curve. And in this individual, a blood pressure of 180 and uncontrolled hypertension. This is brachial blood pressures. But through remote patient monitoring initiation, there is an effect that happens that some can attribute to a placebo effect, that the person knows that they are being watched now, so they make lifestyle changes, but that's still very powerful, right? Because there's a disease modification effect that occurs. Perhaps they're eating better, or they decide to sleep better, or go for walks. But as we also use treatments to treat to goal, we can see that there is an effectiveness, but where also, as time goes on, that we need to adapt the treatment, perhaps change it more frequently, or use adjunctive therapies in order to get blood pressure down and keep it down. So there's also the potential of looking at pressure measurements. We're looking at biomarker measurements and what happens to people's behavior. And here is an example of an uncontrolled hypertensive older gentleman who has obesity, and blood pressure is not changing. And then there's an abrupt drop, right? And I've often said, if you don't understand what's happening to an individual, whether that's patient care or in a trial, just ask them, right? Well, what did you do? And this person started intermittent fasting and could see a near immediate and sustained blood pressure improvement. And that is a biofeedback loop and a biofeedback mechanism to understand individuals' behaviors through the acquisition acquisition and use of digital technologies. So there's, and as we talked about remote patient monitoring, and we've introduced remote physiologic monitoring, right? Are the parameters changing in a way in which the underlying cardiovascular physiology is also changing in a beneficial or adverse way? And so technologies have been developed over many years. And this central aortic blood pressure ambulatory 24-hour ambulatory blood pressure monitoring device that we're measuring blood pressures during the day and maybe once a day. But the blood pressure is changing, much like the pressure in any object is changing as it is being used. The blood pressure in the human body is also changing. So through multiple measurements over a 24-hour period, and also at night, very difficult for an individual to wake up and measure their blood pressure, right? A cuff device. But to do it passively using ambulatory blood pressure monitoring to identify risk patterns. In this case was nocturnal blood pressure dipping. All of our blood pressures go down at night. It tells us that the arteries can relax. And if an artery relaxes, if a balloon relaxes, the pressure goes down. That's a beneficial feature that can now be measured. The opposite is equally true. If blood pressure does not go down at night, or there's a reverse dipping, it actually goes up. So it might portend a lot of stiffness in those arteries. When it should be relaxing, it is not, and blood pressure is going up. And in that cohort, they're at a very high risk of cardiovascular events, specifically stroke. So the relationship between arterial stiffness and atherosclerosis, that's plaque. One cannot have a discussion regarding arterial stiffness without understanding that plaque, right? Plaque is a major factor in arterial stiffness, and arterial stiffness itself can lead to plaque. This is an example of a CAT scan of a heart on the left. If the heart looks round, it's gray because it's muscle and blood. It sits next to the lungs, which are black around it. And the spine and the ribs are white because there's calcium there. Bone and teeth is hard. And my cursor is showing it correctly here, that see this gray line? This is a healthy artery, a healthy pipe. It looks gray. You look in this individual, there's a little bit of white. That's a little bit of plaque. More plaque, a lot of plaque. And as now taking forward what we have discussed, that as atherosclerosis continues, its burden increases, there's a lot more stiffness in those arteries. That stiffness can lead to more pressure over time. So the measurement of arterial stiffness is a physiologic correlate, a biomarker of the underlying health of the arterial system. And so we've talked about device-based measurements. We've looked at biomarkers, right, for analytical and clinical validation. And again, the first slide that I discussed was there's a lot of data on people. We're just not using it. So employing methods such as patient similarity. Now, all of us here and in the audience, we all look different, we are different, but we also have some similar features, right? Our blood pressure waveform has a similar feature. Our ECG waveforms have similar features, although, and the differences tell us something about what is happening in underlying physiology between you and another individual. And so precision phenotyping using real-world data, that's electronic health record data, that's imaging data, ECG data, to now employ what has been done in genomics research of patient similarity networks to identify subtypes and clusters of individuals in relationships to an outcome. So if we look at this animation, this was that, we are all somewhat different, we are all somewhat similar. And if we cluster these features, we can create a map of a disease trajectory using topographical data analyses and other computational methods to look at the range of mild to severe disease. And so we largely look at a disease today as it is mild, intermediate, or severe. But we know that there are variations within these categories and there are a relationship between them. And so looking at precision phenotyping in real-world evidence clinical trials, in this example of using electronic health record data, of all available data, demographic data, of treatment characteristics data, of biometric data, to start understanding clusters of who are smokers, diabetics, hypertensive, coronary artery disease, and now look at remote monitoring. Use a suitable device or a right device that marries that person's underlying cardiovascular risk with what we can do with those biomeasurements, and now look at outcomes in a more individualized approach. And so no discussion about either digital or clinical trials would be complete without diversity, equity, and inclusion. And we've known these challenges over time, but they have been magnified and highlighted during the COVID pandemic, where a lot of the medical innovations that were used left people behind, those of different socioeconomic classes, geographies, and disabilities. And so my apologies. And so we had experience of using digital health technologies at the point of care in very resource limited areas of low and middle income countries and across the United States on the Appalachian Trail in West Virginia, where people commute 200 miles to see a primary care physician. The burden of cardiovascular disease in West Virginia is actually higher than it is in India. in border clinics with Mexico, here in California, and then in desert locations, across rural and desert locations across the United States, where can we use these technologies in a responsible way in order to understand and capture an individual's cardiovascular disease and their risk remotely, and now start enrolling into clinical trials and for follow-up. And so given common goals, current technology could support a common information base that can support the primary mission, better outcomes for patients. So we have data, we are using it, looking at physiologic measurements and arterial biomarkers, precision phenotyping, and with equity, we can then look at transformational methods, whether all at one time or one at a time, to start understanding how novel treatments can be used, their safety and effectiveness in real-world evidence designs. So I thank you for your attention. Please keep in touch, and I look forward to our Q&A.
Excellent. Thank you so much, Dr. Bhavnani, for the presentation. As you mentioned, we are now ready to begin the Q&A portion of the event. And as we do so, I invite the audience to continue sending either questions or comments. Just a reminder, if you're attending via GoToWebinar, please use the questions chat box. And for those of you watching on LinkedIn Live events, feel free to post your questions using the comments tab, and we'll try to get through as many as we can in the time that we have left. Just a note, if we don't have a chance to get through your question today, you'll absolutely have a chance to follow up. We'll share that contact information again, and the team will have a chance to reach out to you after the webinar today. I'm going to go ahead and start off with the first question that's come in. We've had a few come in so far. This question really interesting, and one that I've been kind of wondering about myself. In a world now where wearable sensors are becoming more common, people have their Apple Watches and their Fitbits and all these devices. Do you think this plays any role at all in patients' stress levels about their own health? Especially you can see these results 24/7. What do you think? What do you think kind of effect it's having on patients and their own relationship with their health?
I mean, it's a very, very good question, right? And for those and the individuals who have posted this question, they may have experienced it firsthand, right? It's multifold, right? Are we understanding that individual's health or their disease if we're not also understanding their stress or the overall burden that they have in the real world? So it is a very pointed question, but a very valuable one, where through these measurements and the analysis of these measurements, can we start understanding new patterns of health and new patterns of disease? I'll give an example here, and one outside of cardiovascular now, that an activity monitor is largely used to hopefully make people more active. It's debatable if that actually happens, and for those of you who use one, I wonder if it sustained your activity improvement, but for many people it does not, right? So there's a novelty in it, but there's also a behavior pattern in it. Some people may start to become more active, but they just can't sustain it, versus those that never do. So there is, can we target these individuals better? But the opposite now, if an activity monitor is given to a cancer patient who's undergoing chemotherapy, right? The purpose is not to make them more active. That's hard, right? When you're undergoing chemotherapy. But what would happen if activity parameters fell off and they're walking 50 steps a day for 5 days? You'd be worried about that person. Did they fall sick? Did they have side effects? Did they get the flu? Are they not eating? Are they not? So looking at the opposite, and to your question here, right, it is ripe for data collection, and particularly in trials, right, to try to understand patients' behaviors as they undergo a therapy and they're measuring something through a digital technology. But I hope that answered your question.
Great. Excellent. Thank you for your insight there. Another question that's come in here, and maybe a bit more technical one. This audience member was just wondering if you could describe, you were talking a little bit about intermittent fasting. If you can maybe just describe that in a bit more detail. They were looking for a bit more info on that.
Sure. Much has been published, through research and also through media on intermittent fasting. And so there are different methods to intermittent fasting, but that largely defined the same way: calorie restriction, right? And so it is caloric restriction for a certain time period in the day. Some typical patterns can be a 16-hour intermittent fast before eating your first meal. And so, or others can use 24-hour fasting, every other day fasting. But that calorie restriction, what it does to the cardiovascular metabolism, remember, the body is using glucose, it's using carbohydrates, it's using cholesterol. It has to do so in a state of fasting. Right? And so it uses only what it has access to, which are stored glucose, stored glycogen, stored fats, and now is exposed to calories that are taken in. And so the history of this, of course, goes back to prehistoric, right? Hunter-gatherers, where the human physiology didn't know where its next meal was coming from, right? And so what it does is it primes metabolism. It makes that the calories that are consumed thereafter more used as fuel rather than stored. And there's quite a bit of data on the cardiovascular benefit of intermittent fasting. And I think one recently published just a couple of weeks ago that showed benefit on cardiovascular health and cardiovascular health, wellness outcomes. And some are looking at can it reverse aging in a way that is beneficial. So a very good question, and if you have any others, please send it our way. But something that is important for people now, they modify their nutrition. That includes intermittent.
Great, excellent. Thank you for that, Sanjeev. I think a little bit of internet lag there at the end, but I think we're all synced up again here. Excellent. We've got lots more questions that have come in from the audience. Thank you to our audience members who have sent questions. We're going to try to get through as many as we can with the time that we have left. Next question that's coming from the audience. This member was just asking if you could provide or share any insights available in the use of the PWA assessment in a pediatric or adolescent population.
That's a great question. I'd be very curious, and for the audience member, if you could provide just a little bit more on what some of your specifics, because I think that's a great question. I do not know the answer. I think in the research areas, there have been studies done in congenital heart disease, perhaps some other conditions, of looking at pulse wave velocities. Most of the data that I'm aware of is in the adult population. But we can certainly go back and take a look and provide any insights that might be there. I would think that given several decades of pulse wave velocity research, whether through longitudinal assessments of risk in individuals, of disease phenotyping, and treatments, I wouldn't be surprised if there was adolescent and pediatric data here.
Great, excellent. Maybe one to follow up on to our audience member who asked that question, reach out and we can keep the conversation going after our live event here today. Another question that's come in here is asking how much incremental prediction response was achieved compared to brachial pressure measurements, and sort of a second component to this question, how did you or how do you or how should we best accommodate for social determinants of health in these kinds of investigations?
Got it. A great question. The first one I'll divide into two. So both prediction and treatment, right? That's the incremental ability of central blood pressure compared to brachial blood pressure on outcome prediction has been validated. And I showed one study. There are several others that while brachial blood pressure in hypertensive individuals is associated with cardiovascular outcomes, so let's say that compared to normal blood pressure, a hypertensive individual may see a 30% increase in cardiovascular outcomes over time. When looking at central blood pressure measurements, if central hypertension is present, either with brachial blood pressure that is high, so both hypertensive, central and brachial, or just central pressure is high and brachial blood pressure is in the normal range, can portend a 2 to 3-fold increase in cardiovascular events over a short time period, approximately 5 years. So there is an incremental prediction from the treatment approach, whether in hypertensive, also in congestive heart failure, coronary artery disease, and peripheral arterial disease. There is a benefit above brachial blood pressure or peripheral blood pressures when reducing central blood pressure measurements with treatment. So it is the hypothesis that a more precise or accurate physiologic measurement of pressure can be incremental in prediction and also treatment effect. Now it is what one does to your second question, need to look at different demographic characteristics as well and different disease severities. So the social determinants of health is of major importance when looking particularly at conditions which are very common. Cardiovascular disease, diabetes, obesity, metabolic conditions, and where clinical trials and even observational trials have clustered, particular socioeconomic classes which are higher. They may not have the diversity of ethnicity in them, many European trials or those done in urban locations. But now when we're thinking and pushing towards real-world evidence, that is the real world of diversity, equity, and inclusion, to make sure that it is representative. Any clinical trial is representative of the U.S. population, but also, make sure that not just a certain minimum of diversity, equity, and inclusion, but to really have adequate representation for looking at the efficacy and effectiveness of a therapy under question.
Excellent. Thank you so much for your answer to that. So more questions that have come in from the audience. This one here, a bit more of a technical question. This audience member is asking about why does central systolic pressure increase linearly in a healthy person? And would it be unhealthy to have a central BP that is kind of below the normal that was shown on your graphs?
Very, very good question, right? Again, for this individual, please send a follow-up, because I had this same question too, as I get a deep understanding of central pressures. It is interesting, right, that brachial blood pressure is not linear over time, right? The curve does have a linear characteristic when we are younger, but when we are older, it has a J-shape curve where it goes up more rapidly. Yet central blood pressure measurements through large observational clinical trials, the ACCT Anglo Cardio Collaborative trial of 10,000 individuals that underwent central blood pressure and pulse wave analysis in these 10,000 individuals between the age of 18 and 90 who were free of cardiovascular disease and didn't take any medications for cardiovascular disease, showed that central blood pressure is linear as time goes, as people age. And this has been validated in several other large observational trials. So it is more so that understanding healthy aging, more precise biomarkers, may give different data, right? And so while brachial blood pressure changes, that is nonlinear, central pressure is linear. And so to your second question, is there on the lower portion?
Yeah, the second part of the question is about unhealthy to have a central BP below the normal that was shown on the graphs.
No, it doesn't seem to portend increased risk of cardiovascular events. It now would ask the question, I put the clinical side on that, why is this person's pressure low? Do they have an underlying condition, right? Do they have valvular heart disease? Do they have heart failure where the heart's not generating, the pressure that it did before? So I think that becomes a marker of understanding a condition. And there's a risk that if a condition is demonstrated, well, that has its own risk. But in general, a lower than normal central blood pressure doesn't portend a higher risk of cardiovascular outcomes, if that is the only reason for why central blood pressures are low.
Great, excellent. And I think your answer to that question kind of serves as a nice segue to another question that we've got here. This question was asking, and you talked about this a little bit throughout the presentation, but maybe we could go for a bit of a refresher on it. This audience member was asking about how central blood pressure, it differs from peripheral blood pressure in terms of its ability to reflect that kind of true cardiovascular risk. And then as an addendum to that, why is that distinction important when it comes to drug development?
Yeah, very good question, right? So, the efficacy of drug development, there are many reasons, but one is accurate understanding accurate underlying physiologies, right? In order to then understand what the treatment effect is, the pre-post effect. And so, using arterial biomarkers and pulse wave analysis to start has been done in a multitude of drug development clinical trials in order to look at safety and effectiveness through central blood pressure and pulse wave analysis. And so much like I said, another example would be understanding glycemic metabolism in diabetes. If using just finger stick blood glucose, there is a pattern, but now when employing continuous glucose measurements, that pattern that informs something more about that underlying diabetes state and how it is modified through a treatment. So both of these examples, pulse wave analysis and blood pressure and continuous glucose measurements, must have rigid analytical and clinical validation, right? Back to that, in order to then look at pre-post investigational treatments. And that can infer a new but more informative pattern, right, of treatment efficacy and then treatment effectiveness compared to conventional measurements, brachial blood pressure, or finger stick glucose. But I hope that answered your question. Is a very good one.
Great, excellent, thank you. Another question here kind of serves as a nice transition as well from your last one. We've talked a lot about the importance of integrating that central blood pressure and pulse wave analysis into practice, but the audience member was wondering if you could just touch upon some of the challenges in actually integrating those measurements into practice, and how can these challenges be addressed to support better regulatory decision making?
Yeah, so integration, right? Integration, I think here is implying how any device measurement is used within the breadth of a participant or a patient's existing healthcare data. And so that is electronic health record integration, that is process integration, that is now adding a lot more data to an expansive, data set in individuals, but it is a very important factor, and it requires a lot of multi-stakeholder collaborations across EHR developers, care institutions, trial organizations, CROs, sponsors, data adjudication committees. The list goes on, but without integration on the other side, patterns can be demonstrated, but understanding risk would be limited, right? Because we're not really looking at the similarities, the differences between trial participants. And so there is a lot of regulatory, national policy related initiatives on doing exactly this, interoperability, making data sets come together. And then the second part is what type of computational approaches can be used when so much data comes together. And so both of these conversations are happening at the regulatory and policy levels. And for those who are interested, we can send some information regarding integration and interoperability.
Great, excellent. Thank you, Sanjeev, for that. I think we've got time for a few more questions here. We'll keep it running. And again, just a reminder to our audience, please continue to send in your questions, even if we don't have time to hit them. We'll have a chance to follow up with you after the webinar, and Sanjeev's email address is currently up there on screen as well that you can have a look at. This next question here that's come in I think is an interesting one. We've talked a lot about, cardiovascular health today and cardio studies, but this audience member was asking to what degree that blood pressure as a measurement, as a marker, could be used outside of cardio clinical trials. For instance, maybe in oncology trials. Is there, how significant of a role does it play outside of the cardio world?
Oh, it plays an equal significance, perhaps if not more, right? And so there have been, and I think my colleague here, Brian, he can also provide some information after this to the audience, that there, drug development in oncology, in diabetes, obesity, neurodegenerative, that to understand not just central blood pressure, but pulse wave analysis. This is taken together. Pressure and arterial health parameters, arterial stiffness parameters. Because if we think about arterial stiffness as not just a cardiovascular phenomenon, stiffness occurs at the level of each organ, right? And that includes cancers, that includes neurodegenerative. And so there are and have been many clinical trials in the drug development area, in academic risk prediction, outside of cardiovascular, in oncology and other conditions. So I'd shown one map of the body like that with some examples there, but the group here can certainly provide more based on the individual's interest.
Excellent. Thank you, Steve. I think we've got time for a couple more questions here before we'll wrap up. This question here that's come in, I think is probably a good one to hit as we're about to close here, just as a nice kind of refresher and a reminder of what you talked about across the course of your presentation. This audience member was just wondering if you could just go back and explain a little bit about why pulse wave analysis helps in assessing arterial stiffness, and why this particular measure is more predictive of cardiovascular outcomes compared to the traditional methods that we're all used to.
Sure. No, great to come roundabout, right, to sort of the basic underpinnings of pulse wave analysis. But when we look at that blood pressure curve, right, the one that I showed with the 2 stars on it, systolic and diastolic, that is a pressure wave, right? And the analysis of that pulse wave analysis, that's the pulse wave, the pressure wave. And so now understanding, right, understanding the forward pressure wave, its reflected pressure wave, and what does the summation of those pressure waves look like, and to analyze that. And so those, now looking at a curve, right, 2 points on a curve, right, that happen over time, so pressure is changing over time, is a marker of how stiff arteries are. So let's look at that, right? If a little bit of arterial stiffness is present, then the pulse wave is going forward, and a little bit is reflecting back. So the summation is a little. But if there's a lot of stiffness, a lot of atherosclerosis, a lot of cardiometabolic dysfunction, right, then that pressure wave that goes forward has to happen much quicker. Remember, blowing through a small straw, you got to really push. So the velocity is increasing, and the reflected wave, the amount of that reflection, is also increasing. So the pressure is augmented even more. So the pulse wave analysis, and these are just some of the measures that we're going over now, pressure and augmentation and stiffness, right? But this is the underpinning of pulse wave analysis, not captured through regular blood pressure devices, through others that look at waveform analysis, as you've seen here. But I hope that answered your question. It's back to the fundamentals of looking at pressure and physiology in the human body.
Yeah, no, I agree. I think that's probably a great place to close it, a great question to end on here. Nice, brings everything full circle, as you said, back to the fundamentals. And personally, you explain it so well, so I think it's great, and I hope our audience found it very insightful and informative. At this point, we have reached the end of our Q&A session. As I mentioned earlier, if we couldn't attend to your questions, if you have any further questions, you can of course get in touch with the team. We've got Sanjeev's email address currently there up on screen, as well as our team at Cardiacs will have a chance to follow up with you after the webinar. I do thank everyone once again for participating. If you're attending via GoToWebinar, I have added a link in the chat box where you'll be able to view the recording of this event, and and you can share this link with your colleagues so that they may register to view it as well. You'll also be receiving a follow-up email from X-Talks with access to this recording. And after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated as it helps us to improve our webinars. Just a note for those of you who are watching on LinkedIn Live, we do encourage you to create an account on xtalks.com and register for this event, which will not only give you access to today's recording, but to our entire library of webinars and other content. Now, I'd just like to thank Sanjeev once more for your excellent presentation. We hope our audience found this insightful and informative, and we wish you all a great rest of your day. Thank you so much, Sanjeev. Appreciate it.
Thank you very much, everybody. Have a nice day.
Take care, everyone.